Yeah, methylene blue, which was a fabric dye.
Yeah.
Yeah, it was a textile dye. And now they find it has profound effects on your mitochondria.
Yep.
Yeah.
This stuff works, man.
There's a lot of stuff that does work, which is very strange because, again, it's profit. When you hear about things that are demonized and that turn out to be effective, you always wonder, well, what is going on here? How is our medical institutions, how have they failed us so that things that do cure you are not promoted because they're not profitable?
Cut to the chase, evidence-based. Pull up a chair, let's get this straight. Peptide buddy, he's your peptide buddy.
As some consider it, methylene blue is a mitochondrial-enhancing, RFK-romancing anti-inflammatory compound, and despite its discovery over a century ago, it takes a couple of people to mention it on the news or the Joe Rogan Experience to spike sales like never before. Methylene blue has even found itself more into the limelight thanks to our Secretary of the U.S. Department of Health and Human Services, also known as RFK, also known as the husband of the wife from Curb Your Enthusiasm. Now, methylene blue is something that's gained public interest in spite of its clinical uses, which have been present for over a century. It was initially isolated in 1876, and for simplicity's sake and to save myself millions of audio retakes, let's refer to this compound, as you'll also see it in the literature, MB. Throughout this video, we won't touch every single piece of literature on the compound. However, we will get into its clinical applications, trials, and neurocognitive disorders, and beyond, and then a big old chunk of data that inspired its recreational use. And of course, all the references I read through to make this video will be cited in the description below. Methylene blue is a highly permeable synthetic compound with a unique structure. It contains a central aromatic thiazine ring system that allows it to carry a delocalized positive charge at neutral pH. This structure gives it special properties as a redox agent. It's often described as an auto-oxidizer because it can undergo repeated reduction oxidation, redox reactions, without being permanently reduced. In other words, it can accept and donate electrons multiple times, cycling between its oxidized and reduced forms, while maintaining its overall function in biological systems. Thus, it does not only serve as an antioxidant, but it's also thought to alleviate impact of conditions which, at their core, lie mitochondrial dysfunction. Let's start off with a dosing caveat that addressing will allow you to best wrap your head around some of the data that you may read on your own. Methylene blue, like other medications out there, is known to have a hormetic dose response, which means its dose response is nonlinear in such a way that smaller doses have different physiologic effects than higher doses. At low doses, it acts as an electron cycler in the mitochondrial electron transport chain, enhancing cellular respiration and contributing to its proposed antioxidant effects as well. Conversely, at higher doses, methylene blue can have inhibitory effects on cellular metabolism, and at a notably high dose margin can cause mitochondrial dysfunction. This biphasic response underscores the importance of research targeting dose optimization and therapeutic applications to harness its beneficial effects while avoiding potential adverse outcomes. Now, MB has a long history of clinical applications, including the treatment of malaria, methemoglobinemia, and carbon monoxide poisoning, as well as use as a histological dye given its vibrant blue hue. And although we already touched on some of these, some of the properties thought to propel the actions of MB include enhancement of mitochondrial function, action as an antioxidant, and inhibition of monoamine oxidase, increasing presence of neurotransmitters like serotonin and dopamine. Clinically, it's FDA approved for the treatment of methemoglobinemia and shown efficacy in managing ifosfamide-induced encephalopathy and refractory vasoplegic shock. So let's hone in on the clinical utility of today. First, methylene blue's primary role and approval lies in its treatment of methemoglobinemia, a blood disorder where hemoglobin cannot efficiently release oxygen. By converting ferric iron back to ferrous iron in hemoglobin proteins, it can rapidly reverse symptoms like cyanosis and hypoxia. This application, which in this context is life-saving, is reserved for severe cases. Paradoxically, high doses can cause methemoglobinemia, a reminder that dosing precision is critical in the clinical atmosphere. And as we already mentioned, MB also has been looked at for use in treatment of ifosfamide-induced encephalopathy, which describes a rare and serious condition of altered mentation after use of a chemotherapeutic agent called ifosfamide, which is used to treat different types of cancers. Methylene Blue inhibits monoamine oxidase and prevents the formation of neurotoxic metabolites that can exacerbate or worsen this condition. Moreover, it's employed in critical care settings to manage vasoplegic shock, particularly when it is refractory to standard vasopressors, as in there's a critical need to raise the patient's blood pressure. It acts by inhibiting nitric oxide synthase and guanylate cyclase, thereby increasing vascular tone and in a way propulsing blood flow in a setting where patients are critically ill and it's absolutely necessary. Something to consider is that perhaps methylene blue could increase blood pressure. Also, as a surgical dye, given its visibility and color, MB can be used as a diagnostic tool in surgical procedures to delineate surgical margins, identify fistulas, and detect leaks. And finally, in the context of malaria, MB has purported anti-malarial properties, particularly against Plasmodium falciparum, due to its ability to interfere with the parasite's redox metabolism and heme detoxification pathways. This ultimately leads to an increase in the parasite's susceptibility to oxidative damage and lethal toxins. Particularly in combination with other anti-malarial drugs, MB can enhance therapeutic outcomes and reduce development of resistance. Some other frontiers of research include neurocognitive disorders. Methylene blue has shown promise in mitigating neurodegeneration and cognitive decline in models of Alzheimer's disease. It's theorized to work in a way by enhancing mitochondrial function, reducing oxidative stress, and decreasing tau protein aggregation. And so some in vitro and in vivo studies have demonstrated MB's ability to improve cognitive function and reduce amyloid beta plaques. So let's pick this apart a bit more. There's a company called TauRx Therapeutics that's evaluated its compound called TRX-0237 in different clinical contexts. However, ultimately all revolving around individuals with dementia, Alzheimer's disease in particular. There have been a series of phase 2 and phase 3 trials on this compound TRX-0237, which is known as a tau aggregation inhibitor, thus proposed to interfere with the structurally aggregating tau proteins present in multiple neurocognitive disease states. And it's been in a way shown to interact with these proteins in preclinical models. But the caveat here is that these researchers are not always directly studying methylene blue. They're actually studying compounds in a way derived from methylene blue, which is very difficult to pronounce, called methylthioninium. You'll see two different compounds mentioned mainly, HMTM and LMTM, which are the same thing. It's a stable, reduced form of methylthioninium moiety. It does appear MB was used in phase 2 dose-finding trials. However, there was a need to find a more stable compound, and so HMTM, or TRX-0237, was developed to avoid dose-dependent absorption limitations that methylene blue possesses, meaning at higher doses, methylene blue's absorption becomes nonlinear, and the body doesn't absorb it efficiently beyond a certain threshold. Essentially, HMTM is a more stable, reduced form that bypasses some of these limitations. But for completeness' sake and for the sake of strengthening our general education on the topic, it's worth getting into these trials. So, the Lucidity trial, as it's called, aimed to evaluate the safety and efficacy of HMTM in Alzheimer's disease patients. This randomized, double-blind, placebo-controlled study consisted of a 12-month blinded phase, followed by a 12-month open-label extension. During the first year, patients received either HMTM at different doses or a placebo, with all participants transitioning to a 16 milligram daily dose of HMTM in the second half, so the second year. Previous research had suggested that 16 milligrams a day was the minimum effective dose to achieve appropriate blood levels. While full published results are still pending, preliminary data presented at conferences do indicate cognitive benefits and decreased disease progression, but since this was done some time ago and we're still awaiting results, I'm going to wait and see what they say before making any definitive conclusions here. It's important to note that two earlier phase 3 trials showed less promising results. These studies found no significant differences in cognitive or functional outcomes between treatment and control groups when this compound was used as an add-on therapy, and side effects included urinary and gastrointestinal symptoms, as well as a dose-dependent decrease in hemoglobin. Interestingly, these initial trials suggested that LMTM might work better as a monotherapy rather than in combination with standard Alzheimer's medications. This observation led to the design of this lucidity trial, which focuses on different dosages in a monotherapy regimen so we can really hone in on these controllable features that we seek to better understand. So the lucidity trial's design, examining discrete dosages as monotherapy, really aims to address the limitations of previous studies and provide more conclusive evidence on HMTM's efficacy in Alzheimer's disease treatment. I know we just spent a lot of time on that, and I hope it didn't put you to sleep, but we're just getting started, and I thought I should possibly make the above even more concise, but I knew some of you in the comments would mention it because you're some of the best scholars. And if you haven't already, hit that like and subscribe button or not. That said, I'm your peptide buddy, and today we're talking about a compound that's not a peptide, but let it be known that I, regardless, am your buddy and appreciate you all. Additionally, in TBI, traumatic brain injury, MB has been shown to have neuroprotective effects in TBI and ischemia models by reducing oxidative stress, inflammation, and apoptosis, also thought to help in maintaining blood-brain barrier integrity and mitochondrial function. Now that we got some of the core features of MB out of the way, let's explore the research evaluating other contexts that have inspired what has become recreational use of the compound. Among proposed neuroprotective factors in the context of health, wellness, and human optimization, MB is thought by some to be an anti-aging agent due to its antioxidant effects and ability to improve mitochondrial function, potentially mitigating age-related cognitive decline and even perhaps skin aging. A good body of literature supports that in part of its mechanism, methylene blue interacts with mitochondrial health. It appears in part to enhance activity of that electron transport chain, which creates a proton gradient and produces ATP, our energy. It's been seen to act as a regenerable antioxidant in the mitochondria. It's thought to improve cellular processes as to provide increased oxygen consumption. And on top of that, there's interplay, as we mentioned, with nitric oxide synthase, which is why it's used as a last resort almost to bring people's blood pressure up during critical illness like shock, and it in some way may also modulate inflammatory response. Now, with regards to proposed nootropic efficacy, I think we already took a deep enough approach to looking at the trials evaluating its use in neurocognitive conditions. However, there's a bit more to look out here. What guides a lot of the nootropic interest, as does that revolving around a multitude of chronic disease states, is the idea we keep touching on, in that mitochondrial dysfunction is a characteristic feature of different disease states. So given the role of mitochondria in neuronal cells carrying out metabolic processes to stimulate things like thought and cognition, a proposed feature to mitigate the effects of brain aging, if you will, would be to improve mitochondrial health. And there are preclinical models that seem to in a way mimic this idea, where rodents improve different features of memory and cognition in the context of certain types of memory and object recognition, for instance. And another clinical trial that's quite interesting is that MB was tested in patients with PTSD, post-traumatic stress disorder, at a study out of the University of Washington. The goal was to evaluate its role in enhancing extinction learning. PTSD is a life-altering, trauma-induced condition often characterized by conditioned, fear-based response to a trigger reminiscent of the trauma, and methylene blue was examined in relation to that instinct. The limitations here are sample size and the slower rate of change among those who took methylene blue, which could affect adherence. However, when looking at the results, people were given either MB or placebo in addition to imaginal exposure therapeutic treatments. For the most part, improvements in different contexts look very similar among both groups, with perhaps a slight edge to the methylene blue group. MB augmented exposure specifically enhanced independent evaluator-rated treatment response and quality of life compared to placebo, which are important metrics. The trial highlights the potential utility of methylene blue for enhancing outcomes in PTSD treatment, though further investigation certainly needed. The most common side effect of the trial was urinary or fecal discoloration due to the ingestion of the dye. Here's another fascinating trial. a randomized crossover study evaluating the use of MB for residual symptoms of bipolar disorder was published in the British Journal of Psychiatry. Now, MB, as you'll see, anecdotally a lot of the times, is implicated in some cases in positive mood changes. And a big reason why it's thought to be is via not only monoamine oxidase inhibition, which would raise serotonin levels, but also because preclinical evaluation has implicated enhanced release of dopamine as well. But in this study in particular, the patients included were stabilized on a mood stabilizer medication. It's also an anti-epileptic called lamotrigine. Although not the primary mechanism, one proposed role of lamotrigine is an ability to inhibit release of the stimulatory neurotransmitter called glutamate. On top of that, this anticonvulsant drug is thought to modulate GABA transmission, although the effects of such are likely different at different doses. And so the suggestion at the core of the study lies in the idea that together methylene blue and lamotrigine may further synergistically inhibit glutamate release, which would theoretically alleviate residual symptoms of bipolar disorder. Moreover, there's a hypothesized mechanism by which inhibiting nitric oxide strengthens neurogenesis, and such, via addition of methylene blue, since it does this, it may in a way contribute to neurological stability. And the results portrayed no statistically significant effects on mania in both the MB and placebo groups. The placebo in this case was a low dose of MB versus the experimental group that got a much higher one. With regards to depression, the group that started on an active dose of MB, the higher dose, had significantly lower depression ratings at the midpoint compared to the placebo first group. There also appeared to be greater reduction in anxiety in the active group. That said, interestingly, there was no change in cognitive effects among the two groups. Now, this is where it's worth addressing a very particular risk, and that is for serotonin syndrome. Different classes of medications like SSRIs and SNRIs, which are typically used in the disorders that this trial shows MB may benefit, depression and anxiety, prevent reuptake of serotonin, increasing the neurotransmitter's presence. MB, which in part acts as a monoamine oxidase inhibitor, does the same. And thus, when people are taking these medications that have long half-lives and increase serotonin, they're at increased risk for a possibly fatal condition called serotonin syndrome, and are likely more prone to it when taking methylene blue. Thus, there exists a special pertinent warning for patients on serotonergic meds considering this compound, making it a necessary conversation to have with a medical provider, if it's something to be considered. This study we just talked about didn't show any concerns for serotonin syndrome, but that said, the subjects were on lamotrigine, a medication that isn't particularly serotonergic, and thus this finding isn't in any way convincing from that standpoint at least. I hope you're not falling asleep because we're not done yet. That said, we're almost there. So next, let's get into a trial that looked at magnetic resonance brain imaging in patients given methylene blue. And the researchers' hypothesis here is that giving a one-time, low-dose oral formulation of MB could affect brain imaging during cognitive tasks. There were not many people enrolled, but the results are as follows. People in the MB group showed an increased boldness of the bilateral anterior and posterior insular cortices during the attention phase of the psychomotor vigilance task, and similarly with activity in the encoding, maintenance, and retrieval neural networks of the delayed match-to-sample task. I know that's a lot of words, but pretty much together these results hint that maybe methylene blue can activate areas of the brain involved in processes of short-term memory and sustained attention, something that hasn't really been looked at previously. And although unconfirmed by larger sample size and evaluations of blood concentrations of methylene blue correlated to particular results, as the researchers portray it, it's a good starting point to build from. I will add that in vitro research in rodent hippocampal cells showed features of enhanced cerebral metabolism by increasing mitochondrial function, oxygen consumption, and glucose uptake in the brain, which likely inspired some of the self-experimentation with MB. However, cell culture research in mice hippocampal cells, of course, does not necessarily translate to human use, especially with regards to complicated dosing. We'll conclude our long discussion on the topic with a look at another topic of interest with regards to human use of methylene blue, and that's on skin aging, given that oxidative stress is certainly contributory to the unpleasing effects of getting old. Researchers used human skin fibroblasts from both healthy donors and patients with progeria, a genetic premature aging disease. The study found that MB was more effective than other antioxidants in stimulating skin fibroblast proliferation and delaying cellular aging. MB did not cause skin irritation even at high concentrations, indicating it's possibly safe for long-term use. In the 3D skin model, MB improved skin health by enhancing viability, promoting wound healing, increasing hydration, and thickening of the dermis. Gene expression analysis showed that MB treatment increased the levels of elastin and collagen, which are crucial for maintaining healthy skin structure. And so overall, the study suggests that methylene blue has significant potential for use in skincare products aimed at reducing the signs of aging, but once again, something that's yet to be verified by a more legitimate, robust trial, which in this case shouldn't be too ethically difficult. Okay, I think by this point we adequately discussed not only the risks and perceived benefits of MB, but we also dove into the bulk of the clinical data that exists, which overlap with this space of health and wellness, optimization, these sorts of things. We also touched on its limitations, as well as some of the notable risks. Methylene blue is a topic of public interest, a growing body of literature and knowledge, yet at this point it's only FDA approved for methemoglobinemia and likely predominantly used off-label for additional support of managing shock in critically ill patients. I hope you enjoyed this video. I tried to make it the most in-depth in this space, looking at as much data as I could comb through without hopefully overburdening you. If you're looking for a further way to support the channel, the best thing to do is hit that like and subscribe button. You can also subscribe to the Patreon to make video requests, get early releases, free guides, and special content. I will add, I just released a BPC 157 educational guide based off available research, the link to which will be in the description below. But most importantly, thank you for your time. I certainly appreciate your viewership, and I hope you have a great day. Take care.
Cut to the chase, evidence-based. Pull up a chair, let's get this straight. Peptide buddy, he's your peptide buddy.